on the 5 important stages of the gene cloning. So we're going to take a look at this figure extracted from Campbell textbook. So we'll look at all the 5 main or primary steps or stages in the cloning method. So the first one is isolate. First, isolate the plasmid. So this is the plasmid or circular DNA that contains 2 useful or important genes. One is antibiotic. Another one is a Lexac gene. Okay, antibiotic can contain more than one antibiotic resistance here. So antibiotic resistance, yeah. And this is the Lexac gene that contains single restriction sites for restriction enzymes. There are various restriction enzymes. Each restriction enzyme is very specific. And the restriction site is actually located at a Lexac gene. Okay. So apart from that, this Lexac codes for enzyme beta-galactosidase. This enzyme beta-galactosidase is important to digest or to hydrolyze lactose into glucose. This is actually very important later in the screening. We have actually learned on the screening method to differentiate between recombinant and non-recombinant DNA by looking at the colors of the colonies. The blue colors means that it contains the glucose. When it contains the glucose, means that this Lexac is intact, it's functional, it's active. It can code for beta-galactosidase and can break down the lactose into glucose. But when we look at the white colonies is recombinant DNA. It means that the plasmid has been inserted with a foreign gene. When it has been inserted with a foreign gene, so that causes the Lexac to be not intact, non-functional, or inactive. So when it's inactive, because of this foreign gene, so it means that it will not encode for the enzyme beta-galactosidase, and the lactose cannot be broken into glucose. So that's why the color of the colonies is white. So that is the screening when it's cultured in the agar plate. Yeah. So remember that the agar plate that is cultured is they put the X-gal, which mimic the lactose. The 2 important components in the X-gal in the agar plate. One is the antibiotic. Another one is the lactose. Lactose or X-gal. X-gal mimic the lactose. So that is isolate the plasmid, and another one isolate the source of gene or DNA source from human cell, for example. So that is isolation. Next is cutting or cleaving. So cut, cleave. So need to use RE or restriction enzyme or restriction endonuclease to cut. The same RE is used to cut the plasmid as well as the source of DNA. So for the source of DNA, there's multiple sites and produce thousands of fragments. But for the plasmid, it only contains single recognition site, so just one site of RE. So you're going to have these fragments, the plasmid, with the sticky ends, both sticky ends. Don't forget the sticky ends for both plasmid and human DNA fragments. Sticky ends means that it contains a single-stranded DNA that can hybridize with the fragments. Okay, for example, like plasmid, it can pair up or complement with the human DNA. It's single-stranded. It's staggered. Sticky end is staggered and is incomplete cut. Next, number 3, then you have to join together. The human DNA fragments and the plasmids join together and form the recombinant. But first, it needs to be sealed using another enzyme, which is DNA ligase. DNA ligase is used to seal the sticky ends of the plasmid as well as the human DNA fragments. So you're going to get this recombinant DNA plasmids here. So that's number 3, productions of recombinant DNA plasmids. Number 4, which is transformation. Okay, so this plasmid needs to be introduced into the host, which is bacteria or E. coli. So one of the characteristics of the host is able to accept the plasmid or vector and able to maintain from generation to generations. So introduce the DNA, the recombinant DNA just now, to the host. So if there's an uptake of the plasmid or recombinant DNA, so then the transformation occurs. Okay, but if there's no uptake of the plasmid, there's no transformation, remember. So there's a lot of possibilities here. Probably there will be uptake of plasmid with recombinant DNA, or there will be uptake of plasmid without the recombinant DNA. Okay, so with recombinant or not recombinant, or another possibility means that it will not take any plasmid. If it doesn't take any plasmid, there's no transformation occurs. Okay. If there's no transformation occurs, so all this bacteria, reproducing bacteria, if it's cultured in agar plate, it will die. Because why in agar plate? If it's cultured, because agar plate contains the antibiotic and NaCl. So the antibiotic will kill this bacteria. Okay, so that's why the plasmid is very important, because it contains antibiotic resistance, so that it can protect from the antibiotic. So it will still survive, it will still reproduce in the agar plate for screening purposes. Okay, next, number 5. Plate the bacteria on agar containing ampicillin and NaCl. This is the screening method. So plate the bacteria. So this agar plate contains the ampicillin, the antibiotics, one type of antibiotics, and NaCl, which resembles the lactose or which mimic lactose. Incubate this until colonies grow. So you're going to have two types of colonies. One blue colonies, another one is the white colonies. So take note again on the white and blue colonies that we have discussed earlier on. White colonies contain the recombinant DNA. So means that the Lexac is not intact, it's not functional. It cannot produce a beta-galactosidase enzymes that can break down the lactose into glucose. But the blue colonies means that there's no recombinant, is a non-recombinant DNA, means that it does not accept any foreign gene, and the Lexac is intact. If it's intact, it's active, and it's functional and can encode for beta-galactosidase, and beta-galactosidase can digest the lactose into glucose. So this glucose actually is represented by the blue colonies. So there's a colony carrying non-recombinant plasmid with intact Lexac gene, and there's a colonies carrying recombinant plasmid with disrupted Lexac gene, which is the white colors. The white colonies contains the disrupted Lexac gene, and the blue colors contains the intact Lexac gene. So we're going to have bacterial clone. So the 5th stage is cloning, amplification, and screening. So these are the main 5 stages of the gene cloning method.